Does this look familiar? This is SARS-CoV-2. It's the virus that causes COVID-19, and it's also the reason why we're currently in a global pandemic. Let's go into depth. On a more defined level, SARS-CoV-2 has a plasma membrane, which encapsulates genetic information in the form of DNA or RNA. Specifically, SARS-CoV-2 is RNA. Embedded in its membrane, it has spiky crowns that bind to the ACE2 receptor on host cells. Taking advantage of the receptor, it fuses its plasma membrane with the cell's membrane and releases its genetic material. The hijacked cell becomes a virus-producing factory when it begins to translate the RNA, generating proteins that can be assembled to build up to 600,000 new viruses. Quick brain break. Rock, paper, scissors, shoot. Don't worry if you didn't win, you can just play back the video. Our next focus is small interfering ribonucleic acid. It sounds really intimidating, so let's use an analogy. Pretend that I'm a chef. When a new order is requested, I refer to this cookbook with all my recipes, similar to how DNA in our cells have all our genetic information. I transcribe this info onto a piece of paper and bring it back to my work table, which is where I translate the instructions into food. siRNA is central to RNA interference, a biological process in which siRNA molecules inhibit translation by degrading the target RNA molecules. Going back to our analogy, this means siRNAs prevent the food from being made by tearing up the piece of paper. But I can't just destroy RNA with my bare hands. A multi-subunit protein complex named RNA-induced silencing complex, or RISC, is activated when it incorporates an siRNA. Picture my hands as siRNAs and these scissors as the protein. The siRNA is necessary because it acts as a template to guide RISC to the appropriate mRNA by recognition through complementary base pairing. Base pairing in RNA means A pairs with U and G pairs with C. The protein then cleaves the RNA, resulting in RNA degradation and gene silencing. Synthetic siRNAs can also participate in RNA interference when introduced into human cells. Connecting the two concepts together, this means that siRNAs could potentially be utilized to combat the pandemic because they can pinpoint viral RNAs from SARS-CoV-2 and initiate their destruction. With modern technology, it is possible to synthetically design specific siRNAs through companies like Thermo Fisher and Integrated DNA Technologies. Because the RNA sequence of SARS-CoV-2 is known, we can create these short stretches of siRNA that are complementary to the virus's RNA. Then, in the cell, the siRNAs will be able to identify the viral RNA and chop them up like green onions. The National Center for Biotechnology Information has stated that intranasal inhalation, like a nose spray, is effective in delivering siRNAs to cells. Of course, it needs a package to keep it safe, so we can use positively charged lipid nanoparticles to balance out the negative charge of the siRNA. Neutralizing it will allow it to travel through the membrane to annihilate harmful RNA. Research is still being conducted to study applications of RNAi and gene silencing. New breakthroughs in science are being made every day, and as the youth, the world is in our hands. The idea of age should never suppress or limit a desire for learning. Thank you.